Good morning, class. Today we will be talking about Vibrio fischeri. Vibrio fischeri is a gram-negative bacillus-shaped bacteria. When gram-staining this bacterium, you get a color of pink or red due to its gram-negative cell wall. Gram-negative cell walls contain a lipid polysaccharide layer and a thinner layer of peptidoglycan. This bacterium belongs to the domain of bacteria, the phylum of Proteobacteria, and it used to belong to the clade of Vibrio. Recent biochemical tests and genetic sequencing show that Vibrio fischeri form a tightly clustered clade, distinct from the other genera. Therefore, the clade has since been reclassified. Alright, phylogenetics. The genus that Vibrio fischeri belongs to is Allovibrio, and some common relatives are Allovibrio logi, Allovibrio salmonicida, and Allovibrio wodanus. Vibrio fischeri was discovered in an aquatic environment. It resides specifically in temperate and subtropical waters. Vibrio fischeri has luminescent properties. It produces light when cells are condensed. Luminescent bacteria produce specific chemical compound autoinducers, which can induce bioluminescence reactions in bacterial cells if present in environment in actual needed concentrations. Vibrio fischeri can live within two different states. There's the free planktonic state, where they live in the ocean or in dispersed areas and feed off of dead decaying matter, or they can live as a symbiote within a host. Vibrio fischeri is generally found within the light organs of the bobtail squid. Then one may ask, why does a bacteria prefer to be consumed by another organism? Well, the real answer is Vibrio fischeri provides luminescence to the squid, which would eliminate the shadow that's caused by the moon, which would make the squid a better predator and less of a prey. In turn, the squid provides nutrition and protection for the bacteria. And this graphic right here is an example of an endosymbiotic relationship where a host takes up a bacterial cell and they both benefit from this mutualistic relationship. Alright, so a little bit on the metabolism of Vibrio fischeri. It's a chemoorganotroph. A chemotroph oxidizes organic and inorganic molecules for energy, and the organotroph portion uses reduced organic molecules. It's also a facultative anaerobe, and it cannot denitrify. Quorum sensing is a communication between cells that organizes the expression of genes from the changes in population density of bacteria. Here's a quick animation of how bacteria come together. The bioluminescence is dependent on cell densities, and also quorum sensing was first identified in Vibrio fischeri. The cells of Vibrio fischeri function in a Lux system. The Lux-I gene synthesizes an autoinducer called AHL, and this autoinducer diffuses outside of the cell. If the cell density of AHL is high outside of the cell, then the autoinducers are forced back into the cell. The autoinducers then bind to Lux-R, regulatory gene, and the Lux-R autoinducer complex activates transcription. This process produces more Lux-I and autoinducers, and it amplifies. After amplification, Lux-A and Lux-B are ultimately responsible for light formation. They produce a molecule called luciferase. Luciferase molecule catalyzes a redox reaction that eventually produces a blue-green light. I know that all that information must be hard to wrap your head around, so here's a visual representation showing a few bacterial cells versus many and how the autoinducers are forced back into the cells once there are so many of them. So why are we even learning about Vibrio fischeri? Well, I'll give you a few reasons to why it's an important bacterium to know about. Vibrio fischeri is the first bacterium found using quorum sensing. It uses endosymbiosis with another animal, and also it makes pretty lights. Thank you so much for listening to my lecture, and I hope you'll have a great day.
Created using Powtoon.